Hey guys, here's another conversation on Darshan Talks. We have a really exciting conversation today. We have a special guest. We have a physician, and I'm going to let her introduce herself. Go ahead.
My name is Amanda Leonberg-Yoo. I'm a clinical nephrologist at the University of Pennsylvania.
This is the Darshan Talks podcast. Regulatory guy, irregular podcast with host Darshan Kulkarni. You can find the show on Twitter at Darshan Talks or the show's website at darshantalks.com.
Thank you so much, Amanda, for joining us. Here's some really exciting things. Just to be clear, before we continue, everything Amanda says is her opinion. It is not the opinion of the University of Pennsylvania. We're just having a conversation, so it's quite possible she'll change her opinion tomorrow based on more information that she gathers. So as a lawyer, I kind of have to throw that out there. And to be clear and in full disclosure, Amanda is related to one of our frequent guests, you, who does who comes on, and that's where the U comes from. But we're really excited to have you. One of the reasons we're having this conversation is to talk about pharmacogenomics. Here's a really easy conversation to start with. Have you ever used pharmacogenomics with your patients?
I have. In very specific situations, there are certain toxicities that occur when there's a genetic mutation or a deletion in which I would not prescribe a medication if the patient had the mutation. So I use it infrequently, but it is something in my armamentarium.
And have you found that to be extremely valuable? What kind of evidence have you found behind that to make those decisions?
So the evidence comes from when we use it, we try to prevent toxicities. When specifically talking about pharmacogenetics or pharmacogenomics, we're talking about prescribing a drug in order to elicit a drug response. And we do have certain metrics, so these are our biomarkers where the drug response can be measured. Mm-hmm. And so if there's, the example I think is most relevant is a medicine used for transplant, in which if you had a genetic mutation of TPMT, this would lead to...
What is TPMT, by the way?
It is...
What does it actually mean? I mean, approximately. We don't need a...
Oh, it's an enzyme that actually inactivates the drug.
Okay.
And so you give a drug called azathioprine.
Right.
It's inactivated by TPMT.
Got it.
If you have a mutation in TPMT, the drug is not inactivated.
Got it.
And it remains in high levels in the body,
right,
causing toxicity.
Right.
So we don't have a biomarker to measure this drug specifically. The biomarker we use is actually a bone marrow suppression. So it causes a bad outcome in the patient.
Yeah.
So before we prescribe the medication, we actually test for the TPMT genotype, and if there's a mutation, we would not prescribe the drug.
Fascinating. So you're already using it every single day. Now, do you have any idea, and I don't know if you do, but any idea of where we discovered that it's the pharmacogenomics that does it, and if the evidence behind it is strong? Or was this more a bunch of doctors got together and they discovered this and they've sort of just stuck to it?
That's a really good question, because the FDA actually recommends this genotyping before use of the medication. With a homozygous variant, the non-functioning allele is...
Okay, you're going to have to slow that down a lot for us.
So in this example, it's an enzyme, so
yeah,
you can measure enzyme activity in a lab.
Right.
And so, in like a basic lab, you could take the enzyme, inactivate it using a gene mutation.
Yeah.
However you do that, I'm not an expert in that.
Sure.
And then you measure the enzymatic response or lack of response.
Right.
And so you correlate that. So it wouldn't necessarily be measuring the human body's response. It would be measuring the enzyme, the TPMT enzyme, in isolation. Okay.
So someone decided. So the FDA recommends it. So I would imagine that the FDA got enough evidence to suggest that it's representative and it's a validated method. Now, in my own experience as a pharmacist, there are loads of things we do which simply are how we've discovered certain drugs work. For example, one of my favorite things is when you've got a patient who has quote unquote failure to thrive, you use Megase. So you use 800 milligrams of Megase. Unfortunately, the amount of FDA approved doses, the FDA approved dose is 20 milligrams. So the question becomes, was that never FDA approved? It was never FDA approved, but it's the standard of care. In the same way, do you see the value of pharmacogenomics and other clinicians using it as for unapproved uses, if you will.
I could see that could happen, and I think that's the danger without evidence.
Interesting.
So, the crux of this question comes from, does the gene variant predict response to the medication?
Right.
And that's what you were asking, and
Correct.
I don't think we have evidence for many drugs.
Yeah.
We also have to consider the interaction of other disease states, other drug-drug interactions
Right.
into this algorithm where we're trying to take a drug and predict a drug response.
Right.
So the way I would envision putting this together, pharmacogenomics would come before delivery of the drug. Delivery of the drug can be modified by, in my world, chronic kidney disease. It can be modified by other medications that the patient's on to lead to the outcome of interest. So it's a little bit too simple to say the pharmacogenomics can certainly drive the drug response. So I think you have to think of it in a nuanced way that requires a lot of information about the prediction of response, essentially.
Yeah. Yeah, it makes sense, and we'll always get a chance to that's why they'll come to you to explain it again. But that's why you get paid the big bucks, and we do podcasts. So here's the question. So do you think that there's a future where, Ku came on a previous podcast, and he talked about how there's value in data. And he talked about this idea that it's essential that we get access to this data because that will improve treatment in the future. And a conversation we had sort of before jumping on this on this recording was this idea of disease progression. So I'd love to hear you talk a little bit more about that, and if you think the science and the value of the science outweighs, say, my personal beliefs around, I don't want my genetic data given out. Should I have to opt out of it, or am I automatically opted in, or should there be something different, if you will?
Mm-hmm. I think the first question you ask about how the precision of disease states is kind of the prequel to the pharmacogenomics question. So how can we provide precise predictions about disease progression in a heterogeneous disease like chronic kidney disease? And that really comes also from marrying the genomics and the other external influences, environment, socioeconomic, toxin exposure. How do all of these things amalgamate together and predict progression?
Right.
This, I think, is a really important step before we even get to pharmacogenomics, because it can help inform and also enrich a genomic study by inclusion of all of these individual data points and trying to parse out exactly what is the gene controlling in terms of the outcome of disease. So pharmacogenomics actually has a little bit of a different skew because you're talking about predicting drug response.
Right.
What I'm really interested in as a clinician is, you have a disease, how do I predict your response to a drug or other treatment algorithm, or how do I predict, progression to kidney failure? So I think that sort of precision medicine is the overarching umbrella in which pharmacogenomics falls underneath. And I think having that framework will also influence how we use genomics, specifically pharmacogenomics, in a clinical practice. We have to have that initial precision worked out of how you're going to respond to the disease state in general based on your personal genetic footprint.
So maybe I just want to make sure I'm understanding you correctly. I think what you're saying is, let's not put the cart before the horse. Mm-hmm. Let's not just try to collect data and hope for the best. Let's first get the data with a purpose, validate the data, and then sort of parse that data to mean more. If that's true, first of all, is that correct?
Yeah, that's exactly correct. And I think, again, speaking as a nephrologist, chronic kidney, it'll be extremely important to have a chronic kidney disease population enriched within the whatever data set you're gathering, because it is such a modifier in terms of genetics and outcomes.
So you see that there being value. It just needs to get to that point. So here's a question that I'm starting to wonder. Like, as Ku pointed out in a previous conversation, people are still going to go ahead and get the genetic testing done. You can't put that genie back in the bottle. If that's true, Stat News did a really interesting article where a patient wrote an article saying, "I wish I had someone to explain this information that I got. If I'm a patient, I came to your office and I said, here's my 23andMe profile." And just to be clear, I'm not picking on 23andMe. They just happen to be the most famous one out there. But here's my 23andMe profile. Could you tell me, and is this, please use this to decide what treatment protocol to use for me. What would your take on that be?
I don't think the strength of the evidence is present yet. I think we need to know how relevant is the gene variant in the population. And I, even as a clinical nephrologist, I would not feel comfortable interpreting this data.
Interesting.
Generally, the genetic tests that we do send out will come back sometimes with very specific gene alleles that I do know are, on the causal pathway for some disease state. But very often I'll get results back saying a genetic variant of undetermined significance.
Right.
And that I don't know how to interpret, nor does anyone else. And so you're left with this unknown about how to, counsel a family
right.
and counsel someone about disease progression with this kind of uninterpretable genetic results.
Now here's an interesting thing. We happen to live in Philadelphia. And in Philadelphia, you've got 3 large academic medical centers. You've got some of the best care in the world. If I happen to be living in rural Pennsylvania and I still did my 23andMe test and I got this back and I'm trying to understand what to do and my closest doctor is 45 minutes away, what should I be doing with this information? Should I just not go get the test?
It's a really good question. And I wonder if the 23andMe folks are thinking about this from a bioethics perspective. We always think about the autonomy of the patient decision, the benefit and the non-harm, but this idea of justice there, where the information is the information given is given in equal access to all, and I think in this case that's what's really violated for 23andMe, because the service is provided, but there's no one to help with interpretation.
So you almost suggest that if they want to give out this data, they need to have counselors.
Mm-hmm. Yeah.
Interesting. So that's going to be fascinating. Do they want to take on that liability?
Right. Exactly. Yeah.
Okay. So go ahead. Did you want to say?
But then, how else but where do you start though? This is a starting point where we are collecting information and we could potentially use it. It's a very powerful tool, genomics. And how do we start without violating these ethical principles so that we have enough resources and bandwidth to use it in a meaningful way? I don't know the answer to that.
Fair enough. Let's flip that question a little bit around. And the FDA has made this recent push where they're saying, we really want real-world evidence. We understand that we can do a double-blinded placebo-controlled study and get all the evidence in that format. But the truth is, most patients miss doses. Most patients have some kind of problem, which is why they couldn't enter your study in the first place. We need to see the messy data. To me, pharmacogenomics is part of the data they're collecting is part of that messy data. If that's true, do you see a value to this type of real-world evidence? And do you see physicians discounting the data because it is muddy, shall we say?
Yeah, that's interesting. So you're getting at the point of, is a pragmatic clinical trial the same as a randomized controlled trial?
Yeah.
I think actually pharmacogenomics may help push the field in terms of rapid throughput of a clinical trial. So by enriching a clinical trial, it would not be real world. You would have to have some baseline genetic or genomic footprint to get placed into the clinical trial. But then you're really testing the proof of, is the drug associated with the outcome of interest. So you could, in my mind, I think it'd be very helpful to enrich these trials to get to the answer faster for the drug companies.
So here's that's a great question, and it's a nice sort of segue, which is I've heard some people say that if I can get access to a patient's genetic information, I can help a clinician drop 1 day of stay because the dosing for that specific drug will be reduced because I know that this patient's an inducer or is not, and that might change what dose I start with, and therefore 1 day of stay for a Coumadin dose or a Tegretol dose is quite expensive, and that's valuable. So what is your take on that?
Yeah, I think that's kind of a nice concrete way of thinking about how pharmacogenomics may be helpful in a clinical setting. So again, going back to the transplant example, so using a medicine we use in transplant a lot, tacrolimus, requires it's a variable drug dosing to elicit a drug response, which in kidney transplant, the drug response is the tacrolimus level.
Right.
Now in this case, we do have a level that we can measure, so that's our biomarker. So if we can use the genomics to modify the drug response, that's great.
Yeah.
There are other modifiers you have to consider though, which would include other medication. I think you still need all these effect modifiers, the delivery volume of distribution of drug, the other non-kidney clearance, the kidney clearance, other medications. It's part of that though, and I think it could be very powerful.
So it's a factor of undetermined value at this moment. Is there anything else you'd want to say before we sign off?
No, I don't think so. Thanks for having me.
Thank you for coming on. Stay tuned. Hopefully we'll have her again. Took a little bit of convincing her to come on this one, but stay tuned. Thanks again, guys.
This is the Darshan Talks podcast, regulatory guy, irregular podcast with host Darshan Kulkarni. You can find the show on Twitter at Darshan Talks or the show's website at darshantalks.com.